Namaskar dosto, welcome to channel Toxicology and more. In this video, we'll be discussing another DABT topic that is Toxin. So this question is taken from ABT recertification question paper year 2009. So the question is, which of the following is not true regarding Toxin? So we need to identify from the below statements which are the statements which are not applicable to Toxin. So right answer first we'll discuss right answer and then discuss the rest of the four options. So the right answer is Toxin. It is concentrated in the reproductive organs of affected fish and occurs at the highest concentration just before spawning. So this particular statement, no, high concentration in the case of reproductive organs and during spawning. This is applicable not to ciguatra fish. It is applicable to tetrodotoxin, that is in the case of puffer fish. So we have discussed about this puffer fish toxicity. So I will give the description in the link in the description. You can see how during the spawning, the concentration of this particular tetrodotoxin increases in the reproductive organs, that is ovaries. So whenever you are cooking this puffer fish, if you remove those toxic reproductive organs, rest of the fish is edible. So this statement is applicable to that type of poisoning. So from this you can understand how the examiner will try to confuse you. You will remember this concept, but that concept is not belonging to this type of fish poisoning. Regarding fish poisoning, only three things you have to remember. One is combined fish poisoning, Sugetra fish poisoning, third one is tetrodotoxin. These are the three things. Other than this, they ask, but these are the repetitive questions which they have asked from the previous exams. Okay, so this one will discuss Ciguatra toxin. So first option, how it is correct, we will see. The first option is toxins are heat stable that are a hazard even in cooked fish. So regarding this one, no, these toxins, no, when you will heat, no, the certain toxins they undergo degradation. For example, in the case of histamine poisoning, that is Scromboid poisoning also, even when you heat the fish, the, the particular toxins are heat stable. Why they are heat stable, that also you have to understand. Why? Because no, during the evolutionary this thing, if you see every species is trying to survive, trying to evolve. So they know how to overcome the heat resistant conditions also. They will develop certain mechanism biologically, and that is the reason even when you are heating this particular type of fish, there are certain toxins they do not degrade. So toxins are heat stable and thus are hazard even in cooked fish. For example, there is a silveter fish, like there are different fishes are there. For example, we have here got a barracuda fish or snapper fish. Even if you are cooking that particular fish, the toxins which are there in that particular fish, they will not degrade even upon heating up to 100 degrees centigrade or above temperature. Okay, that is the key point that you have to remember. Same thing you have to remember with respect to histamine poisoning, also scromboid fish poisoning. Second one, uh, because why I am trying to relate is in the case of milk, if you see pasteurization of milk, it will kill certain microbes, but that same principle cannot be applicable for all type of food poisonings. Okay. Second one is it is produced by marine dinoflagellate and is passed up the food chain from herbivorous fishes to large ciguatra fish. So ciguatra fish poisoning, you have to remember biomagnification. So this concept is very, very important. So this particular dinoflagellates are there. They will release this ciguatra toxin. And this dinoflagellate, they are consumed by the small fish, small herbivorous fish. So for example, we have parrotfish as surgeon fish. Okay. Then the small fishes are consumed by the larger fish, that is predatory fish, that is snapper fish and barracuda fish. Generally, we consume this one. So whatever the concentration was there in the dinoflagellate, it bioamplified. The concentration increased up the above the food chain. And larger ciguatra fish, for example, grouper fish is there, amberjack, barracuda, maniil. So these are the fishes which we are consuming, and these fishes will contain more concentration of ciguatra toxin when compared to the dinoflagellates or the smaller fish. So this is concept you have to remember. So one video I have discussed in my channel regarding biomagnification, bioconcentration, bioamplification, persistent bioaccumulation. So I will give the link in the description. You go through. So biomagnification, toxin concentration increases in higher trophic levels. So not only these toxins, there are certain metals, for example, dimethyl arsine, for example, mercury, that is methyl mercury. So that minimata bay disease which you will discuss, which you will be keep getting in your exam also, no? In that case also, biomagnification took place. So those examples also read, not just about food poisoning, also read, try to remember what are the metals which are involved which are causing biomagnification. Okay. Even DDT also you have to remember. across the food chain, how the concentration is increasing. Coming back to third option, it produces neurotoxicity related to an increase in sodium permeability. So whenever you are reading any toxin, you have to understand what is the mechanism, what is the at the cellular level, what it is going to disrupt. So this particular thing is going to interrupt the mechanism is through voltage gated sodium channel. So related to increase in sodium permeability across membranes leading to depolarization of the nerves. So we'll see the mechanism. This cigarette toxin binds to voltage gated sodium channel. This is your normal sodium channel. Okay, where sodium concentration is there. These are the channel through which sodium will enter into the cell. Okay, on nerve cell membrane and keeps them open. So what is happening is that this, in the case of cigarette poisoning, this cigarette will go and bind to this particular whenever the concentration of cigarette toxin increases, no, at the nerve terminal on the cell membrane, instead of, see, these sodium channels has to be open and closed, they have certain milliseconds will be there. But in the increased concentration of this toxin, this particular sodium channel will remain open. As a result, more flow of sodium ions enters into the cell in the nerve terminal. As a result, firing of neurons take place. So you will get seizures. Okay, you will be having C, results in increased sodium permeability, prolonged depolarization stage. So you have, in the case of this membrane potential, you have different repolarization, depolarization phases will be there. No, this particular phase, that is prolonged depolarization, will be taking place. As it is, the channel will be open only. More number of sodium ions will enter into the cell. As a result, firing of neurons, neurotoxicity, seizures, tremors, all these things will be get. This is the take home that you have to remember. Okay. So, what is the explanation which was given by the previous DABT students? Will see toxin usually accumulates in the skin, head, viscera, and roe of the fish. So these are the major parts of the fish where the concentration of the particular toxin is high. Skin, head, viscera, roe, and fish. Okay. Whereas in the case of liver, kidney, in the case of ovaries, reproductive organs, no, that is puffer fish, that is tetrodotoxin, that you have to remember. Okay. So since we have already discussed important pages with respect to different fish poisonings from the Clayson and other case principles of toxicology, today we'll discuss with respect to only diagram we'll discuss. So whatever we can remember from this key notes for the exam we'll see. So ciguatera toxin is a potent marine toxin responsible for ciguatera fish poisoning. So we have different types of ciguatera fishes are also there. I have given the list and I'll give the link in the description that video. So ciguatera toxin is produced by marine dinoflagellate. So certain times you might get this one also. That is Gambidiscia species. What species? They might give or they might not give in the exam, that grows on algae, coral reef in tropical and subtropical levels. So this is very important. At what part, in which tropical lakes or which lakes this particular fish poisoning is there. So generally what these regulatory agencies, what they do is, they will, if they identify one or two catch of fishes where they find the concentration is high, they will ban the fishes from that particular tropical water body. Okay, so that is also very important. That is Gambidris discus toxicus. This is a species, dinoflagellate species. Then we'll discuss about biomagnification. It is passed up the food chain from the herbivorous fishes to larger toxic fishes. Okay, and heat stable toxins are heat stable. Thus are a hazard even on cooked fish. Okay, and this was about the mechanism of toxicity. That is membrane depolarization. Sodium channel will remain open. So more number of sodium entry into the nerve terminal, neuronal axon. As a result, misfiring of neurons, neurotoxicity, tremors, seizures you will be having. Clinical effects we'll see. Clinical effects, how it is going to affect by consuming this Figetra poisoning fish, how it is going to affect your stomach we'll see. It is going to cause nausea, vomiting, diarrhea, abdominal pain. How it is going to affect the neurotoxicity, since neurotoxicity is your target organ. So it is causing paresthesia, that is tingling and numbness, reversal of hot cold sensation. So one thing they will ask in the exam, one of the option you will be having, reversal of which of the following type of poisoning will cause, or marine fish poisoning will cause reversal of hot cold sensation. The right answer will be ciguatera toxic. In the options they might give you scromboid poisoning, they might give you puffer fish poisoning like that, but the answer will be ciguatera poisoning. Hallmark is hot cold sensation, reversal of hot cold sensation. Okay, next you have myalgia, arthralgia, that is muscle pain. Then you have headache, dizziness, fatigue, and weakness. Okay. Always know for different type of poisonings, you have to remember the characteristic features. Okay. Red skin, flushing, like that, some characteristic points will be there for different types of poisoning. So that should you have to remember so that that will help you in eliminating the options from the exam. Next, symptoms can last from hours to weeks and sometimes recur with alcohol, nuts, and certain fishes. Okay. So what is the regulatory status? So one option with respect to regulatory status. So what happens, these poisons, you cannot avoid them. They will be there. You cannot make them zero. So what we can do is that we can put some limits, some safety limits can be established for this type of thing. You cannot stop people for consuming the fish. But what we can do is that we can make the fish, establish the safety of the fish by establishing certain limits for this type of toxin contents. So this type of fish poisoning, no, as per the option, no, it states that it does not have an allowable tolerance established by the US FDA. Food containing ciguatoxin is banned in the United States. FDA considers ciguatoxin a food adulterant under the Federal Food and Drug Cosmetic Act. So this is very important with from the regulatory point of view. Okay, key points that we have to remember from the exam is toxins are heat stable. Okay, and bioamplification, that is sodium channel depolarization, excessive influx of sodium ions and causing firing of the neurons, causing neurotoxicity, paresthesia, hot cold reversal. Okay, no available limits from the FDA. It is banned. Okay, so these are the different examples of cigarette toxin. So even though the question is taken from the year 2009, as per the recent literature, today we are in 2006, there are certain uh, toxic limits established by the FDA. So I have pools of the data. You can also go through here. As of now, these are the current limits. US FDA has not formally banned cigarette toxin. Maybe at the time of 2009 it was there, but today, as per current literature, established strict legal tolerances. We don't have ADI, but there are some tolerance limit, but instead recommended safe guidance level, that is 0.01 parts per billion for Pacific ciguatoxin 1. As I told you, from which region you are picking up that fish, that is also important. Equivalence to zero point 0.1 parts per billion for Caribbean ciguatoxin. So from which region you are picking that fish is very, very important. Any commercial fish testing above these concentrations or originating from high-risk toxic areas is subjected to regulatory action and removal from the market. So the complete lot from that particular contaminated lake or water body, tropical water body will be excluded from the consumption. Okay, so FDA guidance limit, that is specific toxic limit, Caribbean toxic limit, and regulatory stance. What is the regulatory stance? We'll see. Rather than an absolute blanket ban on this real fish, FDA relies on the seafood. This is the regulatory agency that you have to remember. HACCP, what is that? You go back and check. Guidance requires processors to reject high-risk species or harvest locations known for ciguatera poisoning. Okay, so market restrictions are fishery product confined to contain ciguatoxin exceeding guidance levels are deemed adulterated under the federal law and cannot legally be sold to the consumers. So this is a take home that you have to remember. That was option, last option. At that point of time in year 2009, maybe there were no limits, but as of now, we have certain limits. Not exactly allowable limits, but they have given based on the region. Okay, completely US is not banning, but it is not also giving any limit actually. So hope, thank you so much. Be curious. Hope from this video you were able to learn about cigarette toxin. Important things you have to remember. Different types of fish poisoning. Any question with respect to fish poisoning, if it comes, we will be able to address in the exam. Thank you so much. All the best for your exam.